to introduce today the next speaker, Leeman Cruz, a senior at the University of Arizona, and he loves to hike, which is amazing. And the title of his project is BAAMD, which is Build Amino Acid and Nucleotide Data Frame. It's a bioinformatic resource to facilitate HLA research investigator. Leeman will tell us more.
Thank you so much for that introduction. Let me share my screen. Okay, my project is called BAND, or Build Amino Acid and Nucleotide Data Frame. It is a bioinformatic resource for the programming language R that builds tables or data frames of genetic data of HLA regions of the DNA. The HLA, or human leukocyte antigen system, is a group of genes that encode for cell surface proteins that regulate which cells are targeted by your immune system. Substitutions or groups of substitutions called a motif in HLA genes can have a big effect on the presence or phenotype of many diseases like multiple sclerosis, type 1 diabetes, or rheumatoid arthritis. It is important to the study of relationships between substitutions and disease phenotype to be able to access and compute this genetic data. However, the current database of HLA allele sequences, called the IPD-IMGT HLA database, only offers these sequences in a static human-readable form. The data does not allow querying of loci, cannot be easily read or formatted, and contains inconsistencies in formatting across data files. Here's an example of the formatting of the files on the database. There are many problems with the formatting of the sequence information in this file. For one, the sequence letters are only contained in the top row, unless there's a substitution shown here. Also, the 1,000 nucleotide long sequence is cut into segments of about 75, and you must manually count out each nucleotide from the left-hand side. Additionally, the sequences are in an unstructured text file, which doesn't allow for direct access to the information. In short, while a human may be able to make sense of this information, to a computer it is gibberish. My goal was to develop a bioinformatic tool that confronts these issues in the sequence data by consuming the static nucleotide alignment files and generating tables that allow for complex queries of allelic variants, individual nucleotide positions, and sets of positions known as motifs. I accomplished this by building a set of R functions that extracts nucleotide sequence information from the IPD-IMGT HLA database for all HLA alleles, reformats the data, accounting for idiosyncrasies, and generates an R list object that includes computable alignments for amino acids, cDNA, and gDNA sequences. The difference between cDNA and gDNA being that cDNA is the nucleotide sequence for the coding regions of the DNA, and gDNA is the nucleotide sequence for both the coding and non-coding regions. Previously, Livia Tran, who also worked in my lab, produced the BLAS function as a part of the SHARP R package, which takes amino acid sequence information from the IPD-IMGT HLA database and builds a table that allows for complex queries. Her program only works on the amino acid sequence file because there are inconsistencies in the nucleotide files that require changes to the file handling. For instance, here's an example from my program. The cDNA table needs to be broken up into two separate tables because there are two number lines in the cDNA file, as opposed to the one in the amino acid file, which only has one number line. This is because cDNA contains coding regions which can be directly correlated to amino acid positions for each codon. The first number line corresponds to the cDNA position starting from the first nucleotide and going up by one until the end of the sequence. So the first four nucleotides A, T, G, and G are labeled 1, 2, 3, and 4, respectively. The second number line is the amino acid location of each codon of the nucleotides. So for the first codon, ATG, all of the positions are -24. For the next, they're all -23, and so on. I took Livia's program, BLAST, which only builds one table from amino acid sequence files, and expanded it to incorporate cDNA and gDNA file types, and to output 3 additional tables for a total of 4 tables from 3 sources. Because of the inconsistencies between the file types, I had to add many changes to the program But through combining related steps and other tweaks, I was able to limit the additional lines in the code to 86. Her program uses 323 lines to produce one table. Mine uses 409 lines to produce 4 tables. The IPD IMGT HLA database is a central resource for the HLA and histocompatibility and immunojet genetics community, and is the easiest way to access the sequences of all HLA genes. However, access to these genes, especially in a computational sense, is limited by the poor formatting of the database. By addressing this issue and producing a tool that can process the data into searchable tables, this project can help to accelerate discoveries and facilitate new applications of the data. In the future, I hope to expand on this project by creating tools to help query the tables produced by band, such as functions that will assess the validity of queries and return the requested data from the table. I had a great time working on this project this summer. It was engaging and I learned a lot about programming in R, which I had never used before, as well as how many important roles there are in science, not just in conducting the experiments, but also in producing the tools needed to conduct experiments. And so I'd like to thank Dr. Stephen Mack, Livia Tran, and everyone else at the Mac Lab. I'd also like to thank Dr. Fung and everyone at UCSF for providing such a wonderful opportunity for us, as well as the NIH for funding me. Any questions?
Well done, Lehman. Really interesting. I mean, yeah, lots of hand claps here. Was your program named something, given you got Big Dog, Big Cat, and all the rest of it in Dr. Mack's lab? Was yours the band? You gave it another name.
The name was Band.
Band. Okay. Got it. Okay. I didn't know if it was Band, like revised or Band.
Yeah.
All right. I think the first hand up was Sonali.
Oh, I don't think I was.
Oh, okay. Go ahead. Just go ahead.
Sure. It seems like the work you did and the new way of using the computer to assess these genes is so helpful and needed. Do you and your mentor have planned to distribute it or make it more widely known so that other people can use the standard rather than fumble around?
Yeah, so the way this works is that in the R community, there's a community of programmers called who submit their programs and packages for wider use by the community. And a lot of the packages that you do use rely on packages created by other people. I believe it's called CRAN. And essentially, in order to create the application I made, it relied on pieces of code that my mentor wrote and probably other people wrote and uploaded this community. And hopefully other people write programs that require this genetic information and then use my program in their program. And so this will be released to a wider community, and there is a hope that it will be able to be used in other programs or for other purposes.
All right, Mikey, how about you?
Hi, amazing presentation. One question I have, is this similar to like the NIH's program called BLAST? I don't know if I'm making that comparison, but
I've never actually heard of that. What does it do?
Well, okay, so I only like learned of it last semester from a class, but I think from like listening to your presentation, it kind of does something similar, but not as in depth. So I was just wondering if there's like similarities or differences within it. But
I've never actually heard of that, so I definitely can look it up, get some more information on that, and get back to you.
Okay, thank you.
Barry?
Yeah, so I had a question on I know Python and R are quite similar, but they do different things. So I want to know like why you chose R over Python.
That's a good question. Actually, just started getting into both Python and R. Don't really have much experience in Python, but the main reason, so this was kind of built in R prior to me getting into the project. The original function BLAS that I used to make my program was built in R. And the reason why my mentor chose it is he used to work in Python and then found that he was writing code that was like hundreds of lines long. And then he had a colleague that was using R and did the same thing in like 20 lines. And so the difference for him between R and Python mainly being that R allows a lot less lines and syntax and is a lot quicker and efficient. Yeah.
Efficiency, that's helpful. Writing code. Fantastic, especially when you've got 400 lines of it for your code. Excellent.
Yeah.
Well done. All right. Congratulations, Lehman.
Thank you.